test one. So it makes sense to do them as we go through the modules and hand them in as we go through each test, but you don't have to. I'll take them all at the end.
Okay, so we print them out and physically do them?
I don't care how you do it. You can just do them on your computer if you want, just write the answer on a piece of paper.
Okay.
Whatever works for you.
Okay.
Yeah. Or yeah, you can print it and answer questions. I just need something, and you can do them all. I mean, you can go home tonight and be like, I'm motivated, and bang them all out, and hand them all to me on Thursday. Get them over. You're not going to be more motivated about this class probably than you are right now. Just saying, you know how this is. By week 5, everyone's going to be like, oh God, another test, really? I get tired at the end. I feel like Saturday, okay, so we'll do a few slides here. Bring your case study on Thursday if you want, but you're not. It's a pretty straightforward case study, but I'll help you fill in your case study answers. Bring your test review on Thursday. I'll help you fill in the answers to the short answer questions and all that. If you're willing to work, you'll be successful. I promise. All right, here we go. Blood. So you guys can see here, blood. If you should remember from probably I hope AP1, blood is a connective tissue. It's noteworthy in that it's the only fluid tissue in your body, meaning that it has a water part, a liquid part, and a cellular part dissolved or suspended in that liquid part, like it says there. The liquid part, as you probably are already aware, is called plasma. And we can take a sample, of whole blood, and centrifuge it. Put it in a little spinny thing like this, like a merry-go-round called a centrifuge. You're going to do this in the lab, so that's why I wanted to kind of jump the gun on this. When you go to the lab, whoever you have, if you don't have someone that's giving a really great introduction, you've got this. If you have me, you're going to hear this again on, what is it? Tomorrow. Okay, so I can take some blood, I can put it in a test tube, I can put some anticoagulants in it, and I can put it in a centrifuge, and I can separate it, and you need to know this. I can separate blood into 3 parts. This cartoon, shows the 3 parts. The top part is the liquid part. So centrifugation separates things based on their densities. You didn't know. All right, so when I centrifuge a sample of something, the parts separate based not on their weight, although that does play a role, but it's technically on their densities. Meaning that the heaviest, most dense stuff goes to the bottom, and the least dense stuff is at the top. So I have plasma, which I can see is about 50 to 55% of the sample of whole blood. Plasma is the liquid part. So literally it contains water and dissolved solutes, sodium, potassium, chloride, proteins, on and on it goes. In the middle, I have a very thin layer, and on the sample in the class tomorrow, you can barely see this. You have to hold it up in the light and look for it. It's called the Buffy coat. The Buffy coat contains 2 bits. You want to know these. Everything on this slide, what I'm saying. The Buffy coat contains platelets and whatever. Leukocytes. Leukocytes is a fancy science word for what? There you go. WBC, right? And maybe you already know the prefix leuko means what? So leukemia, right? White cell. So leukocytes and platelets are what I find in the Buffy coat. You can see there it's a very small percentage. And then at the bottom I get the other about approximately 50 to 40%. And it's erythrocytes, and that's fancy for RBCs. Exactly. Red blood cells. The percentage, another thing you're going to want to know. The percentage of red blood cells in a blood sample, as you can see here, is called the hematocrit. Hematocrit. And we'll do a hematocrit determination in the lab as well. You can see there why I would want to know the hematocrit. Low or high red blood cells could indicate a deficiency, tumor, or some other cause. Physically, what do we want to say briefly about blood? This is, obvious stuff. The colors, just so we're on the same page, okay? It's red or bright red. Sometimes people want to think, oh, deoxygenated blood is blue or some darker color, right? Why? The models always show veins in blue, and say, oh, the blood is blue. And if I look on this, I'm like, oh, the veins right here kind of look blue underneath my skin. Why? It's just literally because they're underneath my skin. That's a skin thing. The blood inside of all your blood vessels is either really bright red, oxygenated, which is the only blood you've ever seen, by the way. The minute it exits your body, it oxygenates instantly. You've never seen deoxygenated blood properly unless you were in a lab. Or deoxygenated blood, which is dark, deep red. Like this. I don't see a test question, but it seems like useful information to know the blood pH range. 735 to 745. Slightly alkaline. Slightly alkaline. It just seems like a stupid thing to miss on a test if it was over there, you know what I'm saying? Slightly alkaline. The temperature of your blood is slightly normal than body temperature. So one more thing you didn't know, T sub B is the abbreviation for body temperature, but sub B is for body. Capital T is for temperature. That's in degrees Celsius. So your body temperature, oral temperature in degrees Celsius is 37. And you can see there the volume of blood is between 5 and 6 liters, 4 to 5 liters. Those are averages. It's based on the fact that generally men are larger than women. Liters are pounds are times 2 for the number of liters. 10 to 12 pounds of your body weight is blood. For fun, why do I need a circulatory system? This is a trees, forest for the trees. Have you ever heard this expression? Can't see the forest for the trees. Has anybody ever said that? That's an old expression. I never knew what it meant. I used to hear it all the time. My postdoc thought, forest for the trees finally figured out. Forest for the trees means I'm so busy looking at the details of trees that I forget to step back and have a nice view. Look at the forest. Don't forget to lose sight. Don't lose sight of the big picture. So let's address the big picture now. Let's look at the forest before we look at the trees. So, this is a picture of an underwater animal called a sponge. Does a sponge have a circulatory system? What's a circulatory? Let me start really big. What's a circulatory system? Heart, blood, and blood vessels. Do you have that? Okay, good. Does this have a heart, a blood, and blood vessels? No. It does not have a circulatory system. Why not? What does my circulatory system do for me that this creature is having done another way, such that it doesn't need a circulatory system? Why do I have to have a circulatory system? What are its functions?
Transport nutrients, get rid of waste.
Yeah, right. Transportation. I need to deliver nutrients to peripheral tissues at the same time. From those same tissues, I need to remove waste. I need to have a mechanism by which to transport stuff around my body. Why? How does it work here? This is a thin organism. Its circulatory system is the ocean. Water is moving over it. And it's so thin, how does it get its nutrients in? The D word. Diffusion. How does it get its waste out? Diffusion. Would that work for me? Why not? Yeah, that's one reason, right? Do I want this stuff diffuse in and out of my skin? In extremely limited ways. Certain drugs can get in this way, and generally no. I have barriers to prevent that exact thing from happening, right? I need a circulatory system to do what makes sense. Get nutrients everywhere. At the same time removing waste. At the smallest, teeniest, tiniest level, it is driven by diffusion. But diffusion is not enough. Why? I'm dense. And I'm closed. Alright. Boom. Jot them down. I want to know the three functions, right? It's weird, isn't it? Did you like it back the other way? Yeah. Alright. It's getting dark. I like this better too. Alright. Artificial light sucks too. I was like, okay. Alright. Works for me. Okay, functions. Distribution. Like we said, right? Transporting oxygen to tissues and removing what waste gas? What's the waste gas? Carbon dioxide. Delivering nutrients and removing metabolic wastes, urea and others. Yeah. Hormones as well are distributed and transported this way. Regulation is number 2. What am I regulating? Things like body temperature, concentrations of different things, pH. You mentioned fluid volume and its supports with blood pressure and so on. And then the last one is protection. And so the protection is the job of the white blood cells and the platelets, like it says, I think, yeah. So blood clots, and then the white blood cells are fighting off infections and killing tumor cells and so on like that. Another, general thought, right? Distribution of your blood varies based on environmental conditions, right? Okay. In your lab, you definitely need to be able to identify. I know it's a lab for it. You need to be able to name all these cells. The first lab test is a blood test. And they're going to show you slide, very similar to the tissues you did in AP1, except for now it's only on the blood cells. So it's called a blood smear, specifically a Wright stain, because this is the dye is called Wright. Whatever you did design some dye, whoever designed it. So the little clumpy, little fragmenty looking things that look like garbage or they shouldn't even be there, those are the platelets. The most abundant of the cells, these ones are the red blood cells or erythrocytes. Easy. They typically look like little donuts or something. Why? Are they hollow in the middle? No. How do they look? Oh gosh. Yeah. They look like this, right? So when I turn it up on the side, I'm kind of looking at the top cut. Okay. And then I see several different white blood cells, so they're showing me a couple neutrophils. This is where you're going to have a hard time, is identifying the white blood cells. Lymphocytes, these are the T cells and the B cells. And monocytes. We'll specifically address them in a minute. I'll make a couple observations. The lymphocyte, super unique. First of all, the purple dot inside those cells is what? The purple part inside the cell. That's the nucleus. That's the nucleus. You're never going to miss the lymphocyte. Why not? The nucleus is huge. It's the only cell where the nucleus is essentially the entire cell. All right? Huge nucleus, lymphocyte. None of the other ones have that. The monocyte is relatively easy too, and the reason why is the cell is huge. Monocytes are the biggest. The hard part is distinguishing between the neutrophil, eosinophil, and basophil. And it's not that bad really when you look at it. That's where the discrepancies come. And depending on the slides they put on the test, you can figure it out. But I'll help you point out to you the characters. Okay, so we'll look at the plasma briefly and just say a couple things about it. It's the watery stuff with all the dissolved solutes. Yeah, I don't want to go into too much of this, and I wouldn't ask you to identify these specific things on this particular test, but I'll mention some of these points as they come up later on. We talked about the ions already. What's calcium for? What is calcium regulated? Muscle contraction. Remember that? Calcium, some of you could. Calcium controls skeletal muscle contraction, cardiac muscle contraction. Calcium also responsible for neurotransmitter secretion at the terminals of an axon. This is one of the most functionally active ions we've got. When we were talking about the second messenger systems over here, calcium plays a role in intracellular signaling pathways as well. Calcium is critical. And you could argue which one could you or could you not live without? The answer of course is none could you live without. But in terms of being super functional all over the place, calcium is the one. And then again, not surprising that I do what? Store a ton of it, right? Where do I store it? Yeah, I have to have it. Can't live without it. you remember osmosis? Do you remember osmosis? Okay. This is, we'll talk about this more later too, but I want to mention it now because it's important. Okay. In this scenario, if it was this diffusion, which way would the red dots are the solutes, right? So which way under normal circumstances would the red dots move? Towards B, right? But can they go there? In my body, can solutes very easily just get into and out of cells however they want? No. That's the whole point of the plasma membrane, right? Okay. Sequester things in certain compartments. However, I have to have a balance across the membrane of stuff relative to water. But the solutes can't move. You just got done. So what's going to move instead? The water. It's all it can move now. So in this scenario, which way will the water move? A to B or B to A? The other way, right? From B to A. You're with me on this, right? Such that the space between these molecules evens out to match the space between these molecules, right? So here's only 2, and here there's what? 2, 4, 6. So a bunch of water is going to move from this compartment over here, so these ones can spread out. This is extremely important as well. We'll use this later. That's part of the reason why there's so much stuff dissolved in your blood cells. So that the stuff in the blood plasma equals the stuff inside the cells. So that there's a water balance. Do I want tons and tons of water moving from my bloodstream into my cells? Is this a good thing for my cells? Imagine this compartment is the extracellular fluid, the blood plasma. And this compartment is the inside of the cell. That's how it is. Do I want a bunch of water from my bloodstream moving this way into my cell? What's going to happen to the cell? You did this in Bio 1, right? This is the hypertonic, hypotonic, isotonic. Similarly, I don't want it to be reversed, where this is the extracellular compartment, and this is the intracellular. Do I want water from inside my cells leaking out there? No. But both these scenarios will be. We'll talk more about this later when we get to the fluid balance chapter towards the end. But it all does boil down to something to do with the bloodstream related to the number of molecules on either side. The bottom line is, I have to keep them balanced. Okay. Formed elements. So we did plasma, right? Now we do the formed elements. We said already that there's the erythrocytes, leukocytes, and platelets. These shouldn't be indented like this underneath. It makes it look like this has to do with platelets, but it doesn't. Only the white blood cells are truly living cells. Okay? Platelets are small little fragments of the cell. And red blood cells are not really living, they're alive, kind of. They don't have any organelles anymore. We'll look at that in a minute too. So red blood cells have no nucleus, no Golgi, no ER, nothing like that. All of them, as you probably know from Bio AP1, are renewed or born in the bone marrow. Red bone marrow. Okay. We've already made this observation, I think, platelets, leukocytes, erythrocytes. If I look specifically at the white blood cells, I notice that they fall into 2 broad categories, yes? Granulocytes and agranulocytes. What do those words mean? There you go. Okay. So granulocyte is a cell that has granules inside, and agranulocytes, no granules. Okay, you see the breakdown. I mentioned to you already how leukocytes and monocytes are easy to identify, and it's the basophil, eosinophil, neutrophil thing that will have a little harder time. Okay, I do believe we'll stop at 7:30, so give me like 5 more minutes. I'll finish early on Thursday for sure. I need you to learn these vocabulary words for multiple choice questions. Okay? So the first one is hematopoiesis. Don't get confused. Hematopoiesis is the formation of all blood cells, all of them. Either way. there's typically some overlap. So hematopoiesis is the formation of all of the blood cells. And then from there we'll have some other, they all end in oasis, and then there'll be an oasis for the production of each one. So erythropoiesis is for red cells, leukopoiesis for white cells, thrombopoiesis for platelets, like that. The stem cell is called a hemocytoblast. You might remember, maybe, was that maybe? No. Hemo is the blood thing. You know, cyto means cell. And do you remember blast? Means a progenitor. That's where osteoblast, right? Did you guys do myoblast? Okay. Fibroblast. There we go. Chondroblast. Yeah. Just cartoons for this for fun. You know, I don't want to go through this. We're showing red marrow. You know, the cells mature in the marrow. They enter into circulation. We'll talk more about the different types of blood vessels later. Okay, so red cells specifically now, erythrocytes. Once again, we said a second ago the word erythropoiesis is the formation of red cells only. And I know for sure there's a short answer question about this, and so we'll stop at the end of this, okay? Do you remember from AP1 the word complementary? Did you guys learn that? I know I harp on that quite a bit. So if you're not familiar with the topic, complementary, so you can write it down if you don't remember, or if you've never heard it before. Run out of space. Over here. I'll write it over on the other side too. It's complementary. Complementary. Why so important? This word means structures determine functions. The reason I like it so much is it's a way of thinking. You can think this stuff out. And it basically describes the entire course, both AP1 and AP2. So anatomy, the word literally means structure. Physiology, the word literally means function. The entire course, both semesters, all we do is talk about structures and what they do. We talk about the anatomy, and we talk about the physiology. And this word is a one-word encompassing of those ideas. So it means how the structures determine the functions. Or I can say it in reverse. What something can do, its function, is derived from the stuff it's made out of, its structure. So you're with me on what the word means, right? And this is not only true of me and my body, but it's true of my phone, right? LG, LTE network and all this crap and 12 megapixel whatever cameras and all this nonsense. All the cool stuff my phone does is because the little parts it's built out of, right? That's why everyone needs the new one every time it comes out and all this, and I have to condit into needing the newest gizmo or whatever it is, you know what I'm saying? Structures determine function. And that is especially true and an interesting story as it relates to the red blood cell. So, first thing I want to say is what's the structure of the red blood cell? And the structure is suggested on this slide. One, it is a anucleate cell, means there's no nucleus and no organelles. That's a structural observation. What's inside of it? Nothing. Enzymes. Some enzymes make ATP by glycolysis. And what protein to transport oxygen? Hemoglobin. So why is there no nucleus? Why is there no ER? Why is there no Golgi? It's a waste of space. This cell is extremely specialized to do one thing only: transport oxygen. That's its function, right? Its function is, you know, I think, transport oxygen. What structures facilitate that function? One is there's no organelles, which means there's no space dedicated to anything but hemoglobin and a few enzymes to make ATP by glycolysis. That's it. What else? Its shape. is called biconcave disc. So it's two dips on each side. What does this do to the surface area? Makes it go up. That's the idea here. Why has it got a funky shape like this? It increases the surface area significantly. Increasing surface area helps to increase what? That happens in the lungs. Yeah, gas exchange. So the more surface area, the easier it is for oxygen to get in and carbon dioxide to get out. Fast. You get that here, right? So the structure again, to support this gas transportation thing, let's make a shape, have lots of surface area so I can exchange oxygen really fast. Okay, and this slide basically says the things I was just saying. It's all hemoglobin, basically. They only make ATP by glycolysis. Why does that matter? Here's my last thought, and you're all to do some homework for anatomy, right? Why does it matter? I can make ATP two ways, basically. Glycolysis, and what's the other way? Do you remember the other cycle? Krebs cycle, typically I don't do that. Humans don't, we don't ferment, but I can do lactic acid fermentation, but that's not ideal. Glycolysis, and what happens after glycolysis? Bio-1. No? Cell respiration? Okay. No? Oh gosh. What organelle? What organelle for cell respiration? Okay. Mitochondria. Do red blood cells have mitochondria? No. So I usually try to sell Bio-1 like this. None of you have Bio-1, so it's late in the game to sell it. Bio-1's kind of dry, right? Why do I care about Bio-1? Because it applies now. The cool, interesting stuff that you want to learn about now is based in Bio-1. That's why Bio-1 comes before anatomy. You want to know the basics first. Let's apply some basics. In order to make ATP, most of my cells have mitochondria, and I do cell respiration, and I get a ton of ATP this way, right? But it needs oxygen to do it. Yeah? Okay. Red blood cells don't do cell respiration. They only do the first part of that pathway, which is called glycolysis. I don't get very much ATP that way. But I'm willing to make that trade-off because what's the job of a red blood cell? Transport oxygen, right? So it would be kind of a really inefficient waste if my cell that was designed to do nothing but transport oxygen was consuming oxygen in the process, yes? I usually make the analogy like this. If I'm a person who delivers pizzas, and I have to eat pizza to deliver the pizza, I'm not going to be in business very long, right? I'd have to carry way more pizzas to feed my pizza thing to deliver you a pizza. It's inefficient, right? So how do I offset this inefficiency? I have a cell. God made it, or aliens or evolution made it like this, right? It's cool. It's a cool observation. This cell is special. All the other cells have organelles and a nucleus and all these other pieces except for this one. During the maturation process, the hemocytoblast, which has all those organelles, spits them all out at the end. The last thing it does before it becomes a red blood cell is it sheds all of its organelles. So it can do one thing really well. Transport oxygen to everybody else. Cool, right? Woo! Okay. I think there's a multiple choice question about this. I'm 100% sure there's a multiple choice question about this. It just popped in my mind. You can write this one down. This is a no-brainer, but let's not get it wrong. How many oxygen molecules can one hemoglobin protein carry? 4. Okay? Here's how it looks. Here's the hemoglobin protein. Hemoglobin protein has 4 subunits. You see the 4 subunits, yes? 1, 2, 3, 4. And if I look careful, I notice that in the middle of each of these globin, it's called hemoglobin because it's composed of this thing called a heme ring. And the brown stuff here, that's the globin part. The globin part is amino acids. The heme ring, as you can see, is an organic molecule. And in the middle is an iron. How many irons and heme rings does the whole protein have? 1, 2, 3, 4. Each, listen, don't mess this up. Each heme ring can carry 1. But each hemoglobin protein has 4 heme rings. So the answer to the question, how many oxygens can one hemoglobin protein carry? 4 oxygens.
4 iron.
Yeah, 4 irons, 4 heme rings, 4 whole sodium. It's literally 4 mini proteins that all glue together. You got it, right? Okay. Good night. Oh, if you have any questions, I'll answer them. And please, before you leave, if you've not already, somebody